Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Scott Estes, Head of Cell Line Development from Asimov.
Thank you, Alex, and thank you all of you for your interest in our webinar today. I'm going to walk you through our CHO Edge platform, and this is a new offering that we've brought to the biotech bioprocessing industry to use a rational engineering approach for how we engineer living cells. In our case here with the CHO Edge platform, it's to produce biologics. And what we've done is to combine an interdisciplinary approach at the company where we brought together some of the leading scientists in both the cell line engineering and the synthetic biology field, as well as expertise in software design, machine learning, and omics-driven modeling of metabolism and gene expression. So when you bring this cross-functional team together and add to it that computational power and the beginning to understand and predict how cells behave, this has opened the door for us to advance how we do cell line engineering in a much more directed, rational way. So I'm going to walk you through all the parts of the CHO Edge expression system and how it performs. And generally, what we do is to apply these disparate and interdisciplinary approaches to tackle just not only CHO cells, but also are working to offer platforms in both the viral vector and the RNA therapeutic space as well, combining our expertise in these areas. But if we look under the hood of the CHO Edge platform, you'll see that the engine is based upon a robust CHO K1 GS knockout that we engineered here at Asimov. The starting place was the CHO K1 host cell line that we acquired from the ECAC collection, brought the cells to Asimov where we adapted them to chemically defined media, and then did the engineering to create the glutamine oxytrophs. After a substantial characterization at both the genotypic and the phenotypic level, a robust and promising knockout host cell line was then sent to Charles River for banking. And that GMP cell bank that was created has gone through all the appropriate safety, sterility, and identity testing as outlined in the ICH Q5 guidelines. So this is the foundation of the CHO Edge platform. Because all the engineering was done at Asimov, we have a clear line of sight to the cell line provenance, and all that is part of what we offer clients and customers as we either out-license the technology to them to work in their own labs, or alternatively, if we do it as a fee-for-service at Asimov. So the next piece of the CHO Edge platform is an extensive library of genetic parts. And this range from natural sequences to fully synthetic elements that we've designed at Asimov. And it literally spans thousands of different elements, which we have characterized each individually for their performance within our CHO K1 host. So each one of these elements has different properties as it relates to control of gene expression, whether they're promoter elements, UTRs, or terminator sequences. But what you can see shown in the bars above across a log scale is that by combining these different elements, we have well-characterized tools that allow us to explore a very broad vector design space in terms of the control of gene expression. And we find this to be one of the critical elements of the success of the CHO Edge platform, moving away from the historical norms of the industry of relying on a one-size-fits-all vectorology, which kind of limits your ability to maximize the chance of success. by broadening up your vector toolbox to enable you to explore a variety of different vectors and different expression strategies, it greatly enhances your probability of success in maximizing productivity. The last piece of the CHO Edge platform on the biology side is how we actually integrate the genetic payload that's introduced into our host cells. And that's done using a proprietary hyperactive transposase that was developed at Asimov and is delivered at the time of electroporation with the expression vectors in the form of an mRNA encoding this transposase. So just like all the other transposases on the market, the advantage here is the integration of multiple copies in stable transcriptionally permissive sites in the chromatin. So this enables us to routinely achieve highly productive as well as very stable cell lines, as you'll see in a moment or two. Now, if we move to the computational side of the house, all this complexity that's inherent in our genetic library of parts would be really difficult to manage without the benefit of a software tool. And so what we have is a computer-aided design software that we call kernel, which is the repository for our genetic parts library. And with that, the end user can interface with this parts library to build various different permutations of expression vectors with different types of elements and predict and simulate the expression of the desired transgenes dependent upon which genetic elements are used. So that's one of the major advantages of the kernel software, is the ability to manage all that complexity and also to predict expression. But at the same time, other elements of kernel, which are not going to get into today because of the interest in time, but are equally as valuable, are proprietary algorithms around codon optimization as well as signal peptide prediction. So Asimov has used our expertise to develop superior algorithms for both of those applications, and that's now all built into kernel and available for clients to access and take advantage of. So, but really the main power here, and again, talking about, one of the key points I hope to drive home today, and that is that being able to explore a broad range of chain ratios and different degrees of attenuation of your selectable marker are the key aspects of ensuring maximum productivity for a recombinant protein. And this is what kernel does. With all of our characterized elements, we can quickly and rapidly identify the various vector permutations, which will give predefined ratios of different chains of interest for either an IgG or a multi-chain bispecific. And so that's kind of illustrated here in the cartoon on the left with the vector simulation, using different vector strategies to express varying degrees of the different chains, going from high-level expression with the dark blue, and then titrating down as you go from lighter shades of blue to white. And this data set here, I hope, sends the message of what I'm trying to say, in the sense that having multiple vector designs is advantageous from a productivity perspective. Because what we show here is the expression of 4 different antibodies using the same 6 expression vectors across all 4 molecules. Each one of these different vectors varies in one degree or another in terms of balancing the chain ratios in one direction or the other, or alternatively, managing the degree of attenuation of the glutamine synthetase expression cassette. And so what you see here is if there is no one vector across these 6 different swim lanes of vectors, which outperformed with all molecules, and in fact, it's quite variable. In the case of the first example, the gray antibody, we see that a balanced chain ratio between the heavy and light chain maximizes productivity, and we also further need to push the stringency of the glutamine selection as far out as possible. In contrast, with the next 2 antibodies, the pink and the purple, each one of those benefited from having an overexpression of the light chain relative to the heavy chain. However, in the case of the purple antibody, we got maximum productivity in vector 6 at the bottom, which had the most attenuated expression of GS, showing that this was a relatively easy to express molecule, which benefited from driving the selection stringency as high as possible to maximize copy number. In contrast with the pink antibody, that's a more difficult to express molecule, and we had to back off on the selection to get maximum productivity by increasing the glutamine synthetase expression slightly. So again, hopefully this takes the message home that being able to vary your vector design and testing a variety of different vector strategies is going to maximize productivity at the end. And what we see here on the right side of this figure is that by maximizing productivity using this approach, we can get to a substantial pool titer of upwards of 6 grams per liter without any cloning out. And so for clients which are interested in accelerating their clinical development timelines, this type of high-level productivity in pools opens the door to that opportunity. Now, the clones, these are all highly respectable high titer clones. We typically take 48 to an amber fed batch screen, top titers in the range of 6 to 11 grams per liter. And then the next slide highlights a product quality advantage of this broad vector design space as it relates to working with molecules based upon multi-chains and multi-specific constructs. So here we explore a much broader vector design space with 14 unique vectors and basically see two populations of pools emerging from these different vector approaches. On one hand, in the bottom right of the graph, we see a collection of vectors that resulted in relatively high titer at the pool stage, but low percentage of the desired heterodimer. So lots of that titer was coming from the undesired, mispaired product-related impurities. In contrast, we have the second set of vectors in the upper left-hand part of the figure, which showed, albeit slightly lower titers at the pool stage, a much higher percentage of the desired product. So when you take those pools, clone out of them, we ended up in a very favorable place with 8 to 11 grams per liter across the top 12 clones, and anywhere between 80 to 90% of that final titer being composed of the desired product. So really a powerful way to maximize the productivity and reduce the appearance of the product-related impurities due to improper chain pairing. Real quickly, expression stability is obviously a key component of what we do in the cell line engineering field. And with the transposase system, we see the expected amount of expression stability across 4 different molecules, multiple clones per molecule. So at this point, based upon the data set we have taken expression stability off the critical path, as we've consistently seen very stable cell lines coming from this type of expression system. And in the last minute or two, just to wrap it up, I also would like to highlight how our process modeling is further enhanced by using these omic-driven modeling systems that we do consistently with each turn of the R&D cycle. So we generate an immense amount of data, both metabolic, transcriptomic, et cetera, that we use to feed our expression models as well as a CHO metabolism model. Each turn of the crank generates more data, which gets fed back into kernel to further constrain the models and make them more predictive. And so in this case, our metabolic model has been used to interrogate the robustness and the performance of our first generation fed batch process, and pointed to 3 media components that were obvious targets for further manipulation. Changes to a couple of those components in the basal and 1 in the feed resulted in higher biomass and a 25% increase in productivity. So we're continuing to iterate in this manner, and as an opportunity for clients to also take advantage of our expertise in this area as well. And so to bring it on home, hopefully I've convinced you that Asimov has a very robust and powerful expression system based upon not only a very robust host and a diverse collection of genetic elements, but a high degree of sophistication in both modeling and further constraining and developing better systems with these models. So to summarize, we have a CHO K1 based system that consistently delivers 6 to 10 grams per liter. We have the predicted expression stability, as one would expect with transposase, enabling us to take that off the critical path. We have a focus on speed, delivering clients RCBs of a stable, high-expressing cell line in 4 months. Also, I didn't get a chance to talk to it, but we do have a food-ate knockout version of our host, which is equally as robust for those of you working with oncology modalities requiring ADCC activity. And lastly, I mentioned the 2 different licensing options. And I'll just end with the fact that we've become quite confident with our system, such that for basic antibodies, we tell clients that we will provide them cell lines producing 4 grams per liter, or it's free. So thank you for your attention, and happy to take questions.
Okay, great. Thanks, Scott. And as a reminder to our audience, you can go ahead and type in your questions for Scott now. So our first question is, does the CHO Edge system come with freedom to operate, or will there be additional licensing obligations?
Well, so Asimov is on a fairly regular basis evaluating the ever-changing IP landscape on that topic and what we've used to make our cell lines and our vectors. And we believe at this time that we have accessed and licensed the appropriate technology at Asimov, and we provide a single license to Asimov clients that we believe gives them the freedom to operate.
And how many different vector designs do you include in a cell line development project?
So that depends on the complexity of the molecule being expressed. For a rather straightforward IgG, we'll typically use roughly 6 different vector permutations to explore the chain ratios and the glutamine synthetase attenuation. However, when we start moving into multi-chain, tetravalent, et cetera, constructs with greater complexity and more chains, we broaden that vector design space to anywhere between 12 to 16 different vectors to maximize our probability of success.
All right, and our next question is, does your platform provide any benefit for single chain molecules?
It does. I mean, clearly the balancing of chains does not become relevant there. But as I mentioned, the kernel software does have other properties that could be beneficial to a single chain molecule, such as the codon optimization, signal peptide prediction, and then, of course, just the overall robustness of the CHO K1 host that we're utilizing.
And how does your titer guarantee work?
Well, as I mentioned, with a standard IgG, we guarantee 4 grams per liter with the top clone. And if we fail to generate that, then we waive the service fee and all commercial rights are free. If, on the other hand, a client is interested in something outside the space of a standard IgG format, we could discuss potential milestones there as well. But obviously, our greatest familiarity and greatest use at this point are with standard IgGs and multispecifics.
All right, well, it looks like we're running a bit short on time. So if we don't get to your questions, we'll be passing them along to Scott, who can follow up with you. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, how do you assess monoclonality of cell lines?
Yeah, that's a very good question, and important one, given the FDA's focus on this issue. So didn't get to that in the webinar, but great question. So we actually have a dual redundancy in how we assure monoclonality. The first comes with the use of an F-site cell printer, in which, if you're not familiar with the F-site, it's an instrument that dispenses single cells in wells, and at the time of dispensing, it images the nozzle of the dispenser. So you get first an image of a single cell being dispensed into the well, but then to give clients the absolute ironclad assurance of monoclonality, we then follow up with the redundant imaging of the plates with the Cellmetric plate scanner. So we have both the image of the single cell being dispensed, followed by images of the resulting wells showing only a single cell. So in that regards, we feel quite confident that our clients can be assured of a high level of assurance of monoclonality.
Okay, well, thank you, Scott.
Thank you, Alex.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.